Welcome, Duke alumni and friends, to this session of the Duke Alumni Forever Learning Institute, an interdisciplinary educational program organized in a set of thematic courses. I'm Stephenson, Director of Professional Development on the Lifelong Learning team. Thank you for joining our program, Food Wellness, Food as Medicine. This program is part of our theme, Food for Thought, which will examine the many facets of our relationships with food through scientific, cultural, policy, and health lenses. Today's program is the fourth session in the Food for Thought series, and we'll explore how what you eat it connects to your holistic wellness. Super excited to welcome Kim Huffman and Chris Newgard. Kim Huffman is an associate professor in the Division of Rheumatology and Immunology and faculty member in the Duke Molecular Physiology Institute. Dr. Huffman's major interest is in clinical translational research, through which she aims to develop lifestyle interventions, exercise and diet, that improve overall health in persons with rheumatoid arthritis or other chronic conditions and with aging. Chris Newgard is the director of the Sarah W. Stedman Nutrition and Metabolism Center and the Duke Molecular Physiology Institute. Dr. Newgard's research applies interdisciplinary tools for understanding of cardiometabolic disease mechanisms, including gene discovery, metabolic engineering, and comprehensive metabolic analysis. Welcome to the Forever Learning Institute, Kim and Christopher.
Thank you, Anne. So yes, as Anne mentioned, I'm Kim Huffman. I was trained in South Carolina at the Medical University of South Carolina after leaving the Clemson Tiger, Clemson University, where the great football team Clemson Tigers play. And at MUSC, I obtained my MD, but also was trained in molecular biology and was really planning to do research in a molecular biology field, but wanted to combine that with my love of exercise and other non-pharmacologic interventions as opposed to pharmaceutical type research. And so I've been doing that for the last almost 25 years here at Duke, and apply that to my patients in rheumatology, rheumatoid arthritis, or osteoarthritis, but it also reaches all sorts of populations, including aging and other chronic diseases. Thanks, Chris.
Yeah, so I'm Chris Newgard, and the first thing I'll say is I'm a Duke alum. So I'm Trinity 78. I came to Duke from New Jersey, like a lot of people do. And just to tell time the Duke way, this was in the era of Gene Banks and Jimmy Spanarkel and Mike Jaminski. That's how we tell time, Duke basketball. And I came to Duke because I wanted to study marine biology. And you all may be familiar with Duke's Marine Lab at Beaufort, North Carolina. And I had the great privilege and pleasure to spend a semester of my junior year at the Duke Marine Lab. That was a program that was available at the time. And, marine biology was sort of a hazy thing for me. I didn't know exactly what that meant. But what it came to mean was marine biochemistry, thanks to a really dynamic, talented professor named Joe Bonaventura, who sadly passed just a few years ago. And Joe taught marine biochemistry in a way that was so compelling that I knew I wanted to do biochemistry for the rest of my career. But as I looked at marine biochemistry, I said, can I really make a living doing marine biochemistry? So taking the principles of biochemistry, I went on after graduating from Duke to do a year pre-doc, if you will, in Copenhagen, Denmark. I'm originally from Denmark, and I worked in the laboratory of my uncle, Jens Juel Holst, at a time when he was discovering a hormone called GLP-1, which is part of these revolutionary medicines that we hopefully will talk a little bit more about in a little bit, like Ozempic and related medicines that are so amazing for weight loss. So I worked in his lab for a year and then came back to the States to work for Roger Unger and Dennis McGarry at UT Southwestern Medical School in Dallas, and I ended up getting my PhD there. And Dennis McGarry ultimately became my PhD mentor. And this is a guy who trained with a biochemist some of you may have heard of, Hans Krebs, a Nobel laureate. And Dr. McGarry, my mentor, just knew metabolism inside and out. I had this wonderful benefit of learning metabolism from him. I completed my PhD, really having focused on metabolism, but it was the time of the revolution of molecular biology. Everybody was cloning anything that wasn't nailed down. And I felt like I needed to get that experience too. So I went to the University of California, San Francisco to do my postdoctoral fellowship, and then came back to Dallas to be on the faculty in 1987 as an assistant professor in biochemistry. And it was there that I met Sandy Williams, a name that many of you will be familiar with, our former dean of the medical school. At that time, Sandy was the chief of cardiology, the cardiology division at UT Southwestern, and we became close friends and collaborators. And so when Sandy moved to Duke to become the dean, he reached out to me and asked me, would I be interested in coming back to Duke, kind of a long circuitous route, back to Duke to lead the Stedman Nutrition Center. That's what it was called at the time. And I accepted that and came back to Duke, met David and Sarah Stedman. I don't know, maybe some of the people on the call know these wonderful people, great benefactors of our activities for many years, very close friends. They passed just a few years ago. And we built the Stedman Center over the course of 10 years to about 75 people and about 10 faculty. Then we had an opportunity to take the Stedman Center and integrate it with two other groups of folks here at Duke, one being the Duke Center for Human Genetics, and the other being a group of clinical investigators that included Kim and her colleagues, Bill and Virginia Krause, the chief of endocrinology, Dave D'Alessio. And we had an opportunity to bring all of that group, the molecular profiling, genomics. We had set up a metabolomics core laboratory by then. We had a cadre of basic scientists working on metabolic control mechanisms, and then having the opportunity to bring in clinical investigators like Kim and her colleagues. That seemed like a golden opportunity. And thankfully, the dean at the time, Nancy Andrews, agreed. And with the help of Mary Klotman, who was then chair of medicine, now the dean of the medical school, what ended up happening is that we took an old tobacco warehouse in downtown Durham, where I'm sitting right now, and Duke did the really smart thing and gutted it, 120,000 square foot facility, and repurposed it as a modern research facility. And we moved in here as the Duke Molecular Physiology Institute, consolidating the Stedman Center, the Center for Human Genetics, and the group of clinical investigators. And that's what we are today, with a major focus on cardiometabolic disease, both from a clinical translational perspective, but also from a basic science perspective. So interesting road. And I'll close just by saying that when I was an undergraduate, maybe the strongest perception of Durham that you could get was that smell of tobacco, that sweet smell of tobacco that you would get as you came into downtown Durham. And I find it amazing and almost ironic that I'm sitting in one of the buildings where tobacco was literally being cured back at that time. And it's been repurposed in such a beautiful way today. So I thought some of you might like that story, since I'm a Duke alum as well. Kim, back to you.
Yeah, Chris, folks from Duke may be interested in knowing that that building is right across from what's now the Durham School of the Arts, but on Duke Street, the old former Durham High School and junior high school. So if anyone is familiar with that area, that's exactly where Chris is sitting now, with the view of the sports complex and the playgrounds across the street. But so Chris, I learned a lot of stuff about you, and I've known you for, for a while, but that was really interesting. And You've been working in this area for quite a while now, mostly at the basic science level, focusing on food and the way nutrition impact overall health. Could you kind of give a broad overview of your work and some of the insights you've gained? And I'm particularly interested in in your discussion of the concept of metabolomics. You alluded to that a few minutes ago, and how that's led to our understanding between connections between common metabolites, things like glucose, and diseases like diabetes, and how you've used that technology to better understand mechanisms, or have you just found some associations?
Yeah, thanks, Kim. Yeah, so as I said earlier, I've been a metabolic researcher my whole career, and I learned all of the traditional tools of enzymology and being able to measure the concentration of metabolites one by one by painstaking enzymatic assays. Great learning experience because it sort of builds a foundation of understanding that it's very hard to get today, but nevertheless painstaking. So when I came to Duke, it was around the time when people were starting to use more sophisticated instrumentation, NMR instruments, and mass spectrometers to be able to provide a more comprehensive view of metabolic space. And the numbers vary a little bit depending on who's counting, but there's something on the order of a couple of tens of thousands of small molecule metabolites in human beings, and these are all engaged in a variety of different metabolic pathways. And it was fascinating to me, and this was happening just as I was moving from Dallas to Duke in 2002, that there might be an opportunity to get in the game early, and set up shop with some mass spectrometers and other instrumentation to be able to really do a comprehensive job of analyzing the metabolic state of people as well as experimental animal models. So we went to work on that and built a metabolomics lab with particular contributions from a gentleman named Bob Stevens and also Olga Ilkieva have just done an amazing job over the years. And Dr. James Bain has also made important contributions. And today, we're able to measure a large array of metabolites by doing what I call static metabolomics. And what I mean by that is you take a blood sample or you take a tissue sample, you apply mass spectrometry, and you can measure hundreds of metabolites in a sample, the concentration of hundreds of metabolites at a moment in time, right? So let's say you give an experimental drug or you feed a particular kind of meal, and you wait a little bit of time for that meal to be absorbed. You can take tissue and blood samples and get uh, a metabolic picture of how the animal or the human is responding. And we've done a lot of both human studies and animal studies, which we'll get to in a moment. The other thing that we've developed is an ability to do metabolic flux. So in this case, what we do is we take the different kinds of substrates that we're interested in. These are foodstuffs like glucose or protein or fatty acids. And we label those foodstuffs with what's called heavy isotopes. So instead of the C12 mass isotoper, we're looking at the C13. Okay, so it's one unit of mass larger than the C12. And our very sophisticated mass spectrometry instruments can detect the C13 compared to the C12 quite readily. And so if we want to know what a research animal or human is doing with a particular metabolic fuel, we can provide it as a stable isotope labeled fuel and follow its metabolism through its various metabolic pathways to end products. And that, in combination with these static profiles, really gives a fantastic view of metabolic status of humans and animals. And so I'm going to close this first little soliloquy by telling you the first kind of study that we did. And that was a very, very simple study. And it was led by a great colleague who has just retired from the Department of Medicine, Laura Svetke, working with me. And what we did very simply was we compared lean, insulin-sensitive, non-diabetic humans, about 80 of them, to obese, insulin-resistant, but also not diabetic humans, okay? And as you may know, in the progression from leanness to full-blown type 2 diabetes, you pass through intermediate stages. And very often in intermediate stages, you become obese, and the insulin in your body no longer works appropriately. That's what we call insulin resistance. And that's a precursor, a harbinger of an ultimate loss of control of glucose concentration in the blood, which is full-blown diabetes. And so what we were doing here is comparing that intermediate precursor stage, obesity and insulin resistance, to lean subjects with no insulin resistance whatsoever. And for the first time, applying this broad metabolic profiling tool to say, what's changing in that comparison that we may have missed with more mundane and focused tools that were used in these kinds of studies in the past. And to my great surprise, what popped out as the most strongly associated cluster of metabolites with the obese and insulin-resistant state was not fatty acids, not lipid-derived metabolites, as you might expect as you move into an obese state, but rather what might have been considered three boring metabolites, the amino acids leucine, valine, and isoleucine, the branched chain amino acids. These were the metabolites most strongly associated with the insulin-resistant obese state compared to the lean state. And what we've done since the time of that observation is push hard to understand what is that telling us about underlying disease mechanisms. And let me pause there because I want Kim to speak as well. And Kim is an MD. If you ever see me around patients, call the police immediately. But Kim is very, very, has a wonderful manner with patients and research subjects. And she's been doing a really, really interesting study with colleagues in Louisiana, and having to do with caloric restriction. Okay, and it's a study called Calorie. And Kim, why don't you tee that up, and I'll come back to discussing some of the mechanistic kind of work that we do.
Yeah, thanks, Chris. Again, learning a lot. So yeah, Calorie is a research study that was done a number of years ago, sponsored by the National Institutes of Health, and in particular, the National Institute on Aging. And essentially, the background was that in almost every animal, except for humans, from yeast to fruit flies to monkeys, if those animals are subjected to a low caloric intake, they can live longer and live longer healthier. And so that's a really tough study to do in humans, but the NIA undertook it in the early 2000s. And there were 3 sites: Pennington, Washington. Pennington is in Baton Rouge, Louisiana, Washington University, and Tufts in Boston. And Duke was the coordinating center. And approximately 200 people enrolled in the study and were randomized to either eating as usual or a 25% reduction in their caloric intake. And the study lasted for 2 years. So 2 years of reducing your caloric intake by 25% if you're in that intervention arm. And participants did it. And people lost weight, as you would imagine. One of the really interesting things was that people, not only they lost weight, and smaller people burn fewer calories, as many people notice, but even when people lost weight, they burned fewer calories when adjusted for their weight. Sort of the idea that I have tried to put forward as an analogy is that if you have tires on your car and you slow the rate of speed of your car, your tires will last longer, right? So, so the resting metabolic rate improved, or resting metabolic rate was reduced. So they were slowed down, which may imply that they could live longer. And also the biggest really interesting thing were reductions in a number of cardiovascular risk markers, things like LDL cholesterol, HDL cholesterol increase, and a number of inflammatory markers that were really pretty profound. And I think what is the most interesting and may have even gotten the most press is that there have been developed outside of Calorie a number of molecular epigenetic metrics of aging and rates of aging. So say I'm going to make a number up, 40, but my physiologic age may be 35, which is great. I should have picked on Chris rather than myself. But also, not only am I 40, I am aging at about half of a year rather than half of a year per year rather than 1 year per year, or for some very unhealthy people, 2 years per year. In the context of Calorie, some of those measures of physiologic age were actually reduced when people reduced their caloric intake, as well as these epigenetic measures of aging rates, implying that people, when they reduced their caloric intake, their aging rate was actually reduced. So it's a very interesting study, and we are continuing. We have samples and data, and we continue to do many analyses to try to better understand how a simple reduction in your calorie intake helps overall health, and as well as aging rates. So, yeah. So, what do you think about that, Chris? In terms of reducing intake, do you think that's always good?
Yeah, so that's a great segue, because what I wanted to recount now is I think everybody's come on to this call. There's a great attendance today because they expect us to tell them exactly what to eat to be completely healthy. And if you're a little bit overweight like me, how to lose weight. Sorry, guys, it ain't that simple. But let us demonstrate that we've given it the good college try. Okay, so over the years, we have studied a variety of different interventions in humans, dietary interventions in obese humans. We have looked at the DASH diet, which was a diet that was developed here by Laura Svetky. I call it the common sense diet: low fat, low sodium, high in fruits and vegetables, and a reasonably effective way to lose weight. We also have looked at the Atkins diet, and we've compared both of those diets to bariatric surgery. And more recently have compared both of those diets to the latest tsunami in obesity treatment, which are these incretin drugs, Ozempic, tirzepatide, Mounjaro, etc. And so we've got a lot of experience in doing some of the metabolic profiling I was talking about earlier, with all these different modes of weight loss. On the flip side, we've also looked at the impact of overnutrition, either in genetic models of obesity where the animals lack something called a leptin, which is a hormone that controls appetite. And when a gene defect occurs in rats, for example, it makes the animals hyperphagic and they become the size of small basketballs. So that's a genetic model of obesity. Or you can also feed animals, rats and mice, on calorically enriched diets. Okay, so we've studied things in both directions. So let me give you a quick summary, picking up for where I was a moment ago, telling you the first time we used this metabolomics tool and took it in to compare obese, insulin resistant to lean insulin sensitive people, the shock for me was that these 3 amino acids were the metabolites most strongly associated with the obese and insulin resistant state. So carry forward, what else have we found? Well, working with Laura Svetky in her WLM, weight loss management project, which used the DASH diet, we looked at 500 subjects at baseline before they underwent weight loss by Laura feeding them this particular diet. And what we found at baseline is that the branched chain amino acids and metabolites related to the branched chain amino acids created by their breakdown at baseline predicted which of the subjects who underwent diet-induced weight loss would have an improvement in insulin sensitivity. So now it's not just an association, it's at baseline a set of metabolites that can predict clinical outcomes. And that, of course, raises the bar. Around the same time, our frenemy at Harvard, Rob Gersten, did studies in the Framingham cohort, and again did profiling of blood at baseline, and then followed subjects out and determined that the same cluster of metabolites that our group had reported on, the branched chain amino acids, at baseline predicted which of the subjects would be had high risk for developing type 2 diabetes. All right, so now we're starting to see that these metabolites aren't just associated, they are prognostic. At baseline, they're prognostic for an intervention outcome. At baseline, they're prognostic for development of new disease phenotypes. And then when we compared all these different interventions for weight loss, the grand champion of, I'm fortunate, sad to say, the grand champion of inducing weight loss, even today, is bariatric surgery, right? And a somewhat drastic procedure, but extremely effective. And when we looked at type 2 obese subjects, type 2 diabetic obese subjects, and compared bariatric surgery to DASH diet-induced weight loss, matched the two groups for amount of weight loss, made those two groups the same, what we found is that the bariatric surgery was much more effective at improving glycemic control, diabetes, and also had a much more profound effect to lower the branched chain amino acids than compared to the dietary intervention. So every which way to Sunday, we're seeing these associations, and we're seeing that these amino acids are prognostic. Okay, so this now gets into what is it that we try to achieve as an institute, the Duke Molecular Physiology Institute, where we have 45 faculty. Some are MDs like Kim, some are PhDs like me. And people like Kim bring us these amazing clinical cohorts in which we can make the kinds of discoveries using our metabolomics tool that I just described. But me, as a PhD basic scientist, I want to know, what does that mean about the mechanism of cardiometabolic disease, or mechanisms? So in the ensuing years, we have been trying to understand, do branched chain amino acids actually contribute to metabolic disease phenotype? And the answer is that we have found multiple examples where that's the case. So for example, if we add branched chain amino acids to the diet, to an energy-rich diet, a high-fat, high-sucrose diet, and we add branched chain amino acids, it makes the insulin resistance worse than if the branched chain amino acids were not added. If we restrict the branched chain amino acids in the diet in genetically obese rats, we improve their insulin sensitivity. And perhaps the most dramatic example that I can tell you is my colleagues, Philip White and Rob McGarrah, young guys that were postdocs in my lab and got annoyed with me and started their own lab here at the DMPI, and they're doing spectacularly well. Working with them, we found that if you take the isolated perfused heart, so Rob has the ability to take the heart out of living rats, perfuse it such that it's still living and beating, and then you can run different metabolites through that heart and see how the heart responds. When he perfused the heart with elevated levels of branched chain amino acids, as found in obesity, it caused the heart to reprogram itself in terms of the phosphoproteome, and it caused the heart to activate a program of protein synthesis, aggressive protein synthesis. And we know that obesity-related cardiometabolic disease can include unhealthy cardiac hypertrophy. The heart grows and it makes it more difficult for it to pump properly. And here we were able to demonstrate that these very metabolites that we're finding associated with disease, we can show a cause and effect kind of relationship between them and an important cardiovascular outcome. So let me pause there, Kim, and jump back in any way you want.
Yeah, well, I have a couple questions. So one is, quite a few years ago when I was exercising a lot, there was this dietary supplement that I could buy at the cycle store called Muscle Milk. And when I looked at the back of it, the ingredients mentioned these branched chain amino acids. And so I see the gym rats, much more rats than I am, really loving these things. What is it? The context, or are these bad actors?
Great question. And I don't think there's any harm in mainlining branched chain amino acids if you're a well-trained athlete like yourself. Uh, the deleterious effects of branched chain amino acids will tend to come in the context of eating diets that are too calorically rich, not necessarily nutritious, leading towards obesity. That's when having extra branched chain amino acids present. And remember, branched chain amino acids are particularly high in dairy and meat, which are often the things that are part of the things that are consumed to create, an obesogenic situation. It's in that context that they can contribute to the severity of insulin resistance and related maladies. Yeah, so that's a good question.
Well, and I'm going to continue along that same line, because I think a lot of people are interested in this. And I think I saw someone in the chat talk about the Atkins diet. And I don't know how much you want to go down the road of someone's named diet, but I know in my family in South Carolina, there's a big push to have high protein, low carbohydrate diets with things like a lot of bacon and sausage, not sausage biscuits, but sausage biscuits without the biscuit, and those sorts of diets. They're convinced that they lose weight with these diets. What do you think that?
Yeah, I mean, you do lose weight, and there's a simple explanation, which is that when you take the carbohydrate component out of the diet, what carbohydrates do is that they restrict the amount of fatty acid catabolism, the amount of fatty acid oxidation that would normally occur. So glucose produces a metabolite called malonyl-CoA, which binds to a key enzyme called CPT1, and that's a rate-limiting enzyme for fatty acid oxidation. So if you're eating lots of sugar, you're going to slow your rate of burning of fat. And so that's a big contributor to why the Atkins diet can be reasonably effective for body weight loss. The problem is staying on it, right? It is very, very difficult to stay on a diet of that nature. And that's true of any dietary intervention for obesity. And that's why bariatric surgery, but even more so, the incretin drugs, Ozempic, tirzepatide, et cetera, are seeming to have such a tremendous impact, because this problem of rebound weight gain, right? It's New Year's Day. You say, gosh darn it, I'm going to do better this year. You start an exercise regimen, you start on a healthier dietary approach, and you lose some weight. And lo and behold, by April, you're rebounding, and that weight is coming back. And that's because what happens is that the weight loss triggers food ingestion signaling from the central nervous system. You get activation of neurons that stimulate appetite, and your body is driven to get back to the set point that you were at when you started the weight loss. Remember, in ancient man, being a little bit fat and hearty was a sign of success, right? You're making it if you have a normal body weight, or maybe even a little extra body weight. And we have control systems that drive us back to that set point very, very powerfully.
And is the reduction in metabolic rate that happens with weight loss also a contributor, as I mentioned with the
Yeah, no, I think, I mean, you know more about that than I do, and I think your discussion of that earlier was great. So yeah.
So, I was wondering if you could talk. I know you've been doing some work with Sarah Armstrong with food insecurity, and I think that the audience here may be really interested in the kind of global implications that work has.
Right. Thank you. And I'm not going to let you go without some discussion of exercise, okay? Because just so everybody knows, Kim is one of the best people in the world at doing human exercise interventions, and we actually have a joint project in that area. So we'll come back to that. But yeah, thanks to a gift from a generous donor named Swan Lee, uh, Sarah Armstrong and I were able to launch a two-pronged investigation into food insecurity. And Sarah is a pediatrician, and she works with underprivileged families here in Durham, and has set up a beautiful study where she's recruited families at risk for food insecurity, and is going to look at outcomes, future obesity in young kids in these families at risk for food insecurity. And basically, half of the group is going to get what is the traditional, and I'll say it, Band-Aid for food insecurity, which is called the WIC benefit. That's basically money, government money, given to the families that are at risk, who go then and spend on the food to feed their families, including at-risk young kids. In Sarah's case, she's going to have another arm where she's going to visit with the families on a regular basis and provide nutritional counseling. So you're not just getting a bolus of money, you're also getting the counseling to to choose well the foods that you're buying with the money that you're getting for more nutritional outcomes. So that's going to be a really interesting comparison of those arms as we go forward and see which of the kids suffer later in life obesity and other cardiometabolic risk phenotypes, because that's the experience. Food insecurity, somewhat counterintuitively, leads to later in life obesity and risk of cardiometabolic disease. So what we've done, me being the PhD and the basic scientist, we've set up an animal model of this. And we're very excited because using a 3-week period of intervention with food insecurity. And what I mean by that is that we feed food sporadically and at a lesser amount than a group of animals that are eating ad lib. Okay, so on Monday, we'll give 20% as much as the ad lib group. On Tuesday, we'll give 40% as much. So it's sporadic. It's always less than what the ad lib fed group is getting, but it's coming in fits and starts, which again is often what families experience in real life food insecurity. And lo and behold, when we do that intervention, 7-8 weeks later, when we study the animals, they're laying down more adipose tissue, they've gained more body weight than the animals that didn't get the food insecurity intervention. So we think that means we've got a preclinical animal model where we can pick apart the mechanisms that may be responsible for those deleterious outcomes. We're very, very excited about that.
And do you think that's because the sort of the body's, from a teleologic perspective, the body's fear of not having food motivates it to get better at storing fuel rather than using it?
Yeah, you know, the mechanism is what we're going to hunt, Kim. And we have the great benefit of a colleague here in the DMPI that you know well, Rana Gupta, who is one of the world's foremost authorities in fat biology, adipose tissue biology. And uh, as I said, one of the outcomes of the food insecurity intervention is that 8 weeks later, our rats are laying down more fat. Now, how is that happening? What is the fat? How is the fat tissue remodeling itself to grow? Um, and Rana is going to analyze this using all the molecular tools that we have, meaning including measuring all the RNA molecules, transcriptomic profiling, doing metabolomic profiling, et cetera. So we hope to understand what's happening to that adipose tissue at a deep molecular level. And that's the opportunity that we have having a preclinical animal model, is that we can be somewhat invasive and take these tissues out to analyze them in a deep molecular sense. So I see your puppy in the background, but I want you to just make a quick comment about the MRA Pac study. Just set that up for us, and I have something to say about that. And then I think we're probably coming down to wanting to spend some time answering some questions. Why don't you go ahead with that?
Yeah, so MRA Pac is another NIH-sponsored study, and I apologize. Someone is strangely knocking on my office. But MRA Pac is an NIH-sponsored study that's being conducted at multiple sites across the U.S. And from a clinical perspective or human social interaction perspective, the humans are at 11 sites. One of those is children, where people are recruited to perform either aerobic exercise training, resistance exercise training, none. And maybe should I stop and let you take it from
Yeah,
there?
you go. You got your hands full. All right. Yeah, I'm going to close because I think we do have a lot of questions. But I'm going to close with something that, you know, I'm 68 years old. I've been doing this for 40 years. And, the beautiful thing is every once in a while, you get hit in the forehead really hard and you learn something completely new. And what happens, and not necessarily because of willful behavior, is that when we're doing biological experiments in animals, we will tend to do the experiments only in one sex. And what's traditional in the biochemistry literature is you buy male rats and you do whatever dietary or other manipulation you're going to do, and you do it in male animals only. And I am guilty of having done that for many years until I got involved in this MRA Pac thing. And the MRA Pac study organizers and planners and the National Institutes of Health insisted everything we do, both in the human work we're going to do and in the animal work we're going to do, the exercise training interventions that we're going to do it in both males and females. Okay, and so this study is a large NIH consortium study that involves 100 faculty members from all over the country. And Kim is part of one of the clinical sites, and I'm one of the chemical analytical sites with our metabolomics group. And so we do this rat study, and the protocol is to exercise rats on on running wheels for 1, 2, 4, or 8 weeks. And then at each of those time points to sacrifice the animals and do a whole body molecular analysis at the RNA level, at the protein level, and at the metabolite level. And what do you think was the number one factor that modulated the responses to the exercise intervention? It was if the animals were male or female. Diametrically different mRNA, protein, metabolite responses in the tissues that we surveyed in response to exercise, male versus female. So that really opened my eyes. And I come back into my own laboratory to work on branched-chain amino acid metabolism and its relationship to cardiometabolic phenotypes. And we have now discovered that females and males metabolize branched-chain amino acids in the liver at completely different rates. The female metabolizes branched-chain amino acids much more actively and aggressively than males do. And we think that this is part of the explanation for why obese females are less susceptible to diabetes and cardiometabolic disease phenotypes, including fatty liver, which is a growing health concern in our country, than males. We think it's because they have a higher rate of branched-chain amino acid metabolism that influences the concentration of another metabolite called threonine, which has an anti-steatotic property, which is to say it prevents the accumulation of fat. So by getting smacked in the forehead by the observations made in the MRA Pac study, I've taken that back into our own work and discovered profound sexual dimorphism in the way branched chain amino acids are handled that I believe influence important disease phenotypes. So I'm having fun. I hope that's coming across.
Yeah, I find the animal differences remarkable. I'm glad you touched on that. The one that I thought was the most interesting was the retention of fat in the female rats. So I see we have a lot of questions about, excuse me, about time restricted eating and feeding. And I know I wanted to, one thing that we didn't get to talk about was a study that Patrick Smith and I are doing at the Stedman Center for, and Patrick Smith is at UNC, but at the Stedman Center for Nutrition and Metabolism. And we have recruited, it's a pilot study, we've recruited older individuals with mild cognitive impairment. So not severe dementia, but mild cognitive impairment. And we have these people going through a counseling intervention that encourages and helps instruct and support them to restrict their intake to eventually, they sort of ramp up to to fast for an 18-hour period a day. And so what is that? 6-hour window, right? So of eating. And what we are seeing is, and this is just not, a handful of people, 10 people or so, but we're seeing improvements in cognition, and those improvements in not all levels of cognition, but what's called executive function, sort of how you make decisions, those kinds of things. And what we're seeing is those improvements are tied to fasting ketone levels. And so I wanted to share that. And Chris, I don't know if we have time for you to add some information about how the ketones tie in with the branched chain amino acids and all of the other really interesting science that our smart colleagues at the DMPI are doing.
Yeah, I think to me primarily introduces the work of your good friend and colleague Debbie Muyo, and you know, what's becoming very clear is ketones, which are the byproduct primarily of fatty acid oxidation, but also can come from amino acid oxidation, including branched chain amino acid oxidation, have very positive effects on the heart. And what these ketones do is makes fatty acid oxidation more efficient in the heart than if they were not present. And interestingly, when are ketones beginning to be elevated? They tend to be elevated under conditions of caloric restriction. Uh, if you fast overnight, you're going to start to see your ketones rising a little bit in the morning. And if you fast for a longer period than that, then the ketones are going to come up more strongly. And it turns out that ketones have a salutary positive effect on the ability of mitochondria in the heart to oxidize fat to the highest level of energetic efficiency. And this is beautiful work, absolutely gorgeous work of a very remarkable technical nature that our colleague Debbie Muyo has done. And I think this is, again, it fits this idea that we have when we make observations, clinical observations, we want to try to move that to a level of mechanistic understanding. And we call that retrotranslation, from the profiling the molecular signatures in human disease cohorts, which are fascinating and very important and give us biomarkers. But that's not enough. We need to, when we see those associations, we need to prosecute that and retro-translate to try to understand underlying mechanisms. And that's what our institute is all about.
To add one more quick note,
Yeah.
and I won't call out a name, but one member of our audience was kind enough to reach out to say that they were in a research study from our group where we instructed people with rheumatoid arthritis to improve their diet and overall exercise patterns. And this person in the audience said it changed their life. So I just wanted to shout out, but also just to kind of mention to people the power of these human clinical research studies, that we learn a lot, but they also help the participants as well. Chris, I saw one question asked about what's the best diet. And they asked about the Mediterranean diet. And I know you mentioned that Loris Fakih's work was what you call DASH was a common sense diet. And I often use this term with my patients. So can you speak to that from your perspective and expertise as well?
Well, I love the Mediterranean diet personally. I mean, I can eat pasta and olive oil and the wonderful things that you get in the grape leaves and all of those wonderful things. I do, lots of seafood in a Mediterranean diet. Generally those things are quite healthy. Laura's diet is one that is supported and promoted by the National Institutes of Health. It's very carefully thought out. Part of that diet was devised, and the reason that Laura was so involved in it, part of it was devised for hypertension. And thus that diet has there's really a focus on keeping a low sodium component in that diet. But beyond that, I call it common sense because lots of fruits, lots of vegetables, staying away by and large from red meat, and limited amount of dairy and certain kinds of dairy. So to this day, that was a diet that was put together for investigation. Gosh, it must be 30 years ago now. The NIH still places it very highly in terms of its dietary recommendations. So I'm not sure what else I can say than that. I want to be. We did not have this session today for you all to walk out with Chris and Kim said this is what you should eat. Life is too culinary life is too interesting to allow that, in my view. One should always try and enjoy, and life is short. Enjoy these things, but being sensible about amount, being sensible about alcohol consumption, and knowing the foods that are very high in fat tends to be a problem. Watching the sodium, a lot of this is common sense. And but what is really interesting now, we live in a time of these incretin drugs, Ozempic, etc. And I've had the opportunity to do a metabolomic study in collaboration with Lilly, looking at their first generation incretin drug, which is Ozempic, and comparing it to tirzepatide, which is the second generation drug. And the difference is Ozempic has one warhead, and that is the hormone GLP-1. And tirzepatide has two active agents, and that is GLP-1 and another incretin hormone called GIP. Well, we compared those two um, uh, uh, molecules in a human study in human diabetic obese subjects, and it's remarkable that the tirzepatide versus the Ozempic both caused weight loss, both caused improvements in lipids and other variables that we look for as harbingers of improved metabolic health. But if you look more deeply, the Ozempic, the dual agent, caused more striking changes in lipids and very large decreases in the branched chain amino acids that I was telling you about, compared to the mono agent, the Ozempic. And so, so there's still a lot to be learned about how this is happening. And I'll just a quick shout out about research that's going on here in our institute. Rana Gupta, again, our adipose specialist, working with a very talented young scientist named John Campbell. They are trying to understand how this GIP component of tirzepatide, the dual agonist agent, how that is actually working on the fat tissue. Because believe it or not, the drug companies who are promoting these dual agents really do not understand exactly how the GIP component is working on fat tissue and other tissues. And my guys here in this institute, without divulging their unpublished information, may have figured out how the GIP component is actually working in adipose. So we're very excited about that too.
Thanks, Chris, especially for helping me with my dog issues. So I just while I'm managing that, and you're managing the chatting. I was scrolling through the real chat, the chat bar, and someone had mentioned what the components of an anti-inflammatory diet are, and this is something I get asked a lot of times in my clinic, and I generally go back to the common sense approach. But what's your sense of changes in inflammatory markers with the GLP-1s and the GIPs?
Yeah, I mean, I think in general, uh, you lose a lot of weight with all of these agents. In particular, with tirzepatide coming along, and not only causing weight loss, but causing a more dramatic improvement in overall metabolic health than the mono agent. Um, Usually, obesity brings with it in lots of inflammation in the adipose tissue and in other places as well. And so, yeah, you see that begin to be moderated. And now, what's interesting is that the drug companies are thinking about adding a third component to these drugs to make it a tri-warhead, and that is the hormone glucagon, and that is likely to increase fatty acid oxidation, so to help with the liver fat issue, and potentially cause even more weight loss than either the mono or the diagonist drugs. So stay tuned and hang on to your hat.
Well, these questions are wonderful. Thank you to Kim and Chris for a great session, and to our audience for joining us today.